Hello everyone. Today we will be discussing on sterilization and disinfection. On this topic, I have decided to put four videos. No, not four, three videos. That will be first will be on introduction of sterilization and disinfection. Then methods of sterilization and disinfection that will be discussed in the second video, and then disinfectants and their role we will be discussing it in the third video. Coming to the first video, that is the lecture on definition. Here I will be defining some certain journal terms used in reference to and which are related to sterilization and disinfection. Then we will be defining sterilization and disinfection also. And then last one will be the difference between sterilization and disinfection. Certain terms which a student should know while studying sterilization and disinfection topic. One of them we start with antiseptic agents. So what are antiseptic agents? These are the chemicals used to remove pathogenic microorganisms, that is the microorganisms that can cause infection, which finally leads to illness. So the removal of these microorganisms is done by certain chemicals, and these chemicals are used for on the external surfaces of the body only. That is like skin. One of the examples is skin. So these chemicals are called antiseptic agents. Another type of chemical is bacteriostatic agent. Now, what is bacteriostatic agent? That is a chemical used to arrest growth and multiplication of bacteria. That means it will make bacteria static. It will not grow and reproduce. Another type of chemical is bactericidal. What is that? That this bactericidal agent is are the chemicals which are used to kill or inactivate bacteria. It will kill the bacteria. So there's a difference between cidal and static. Static will stop reproduction and multiplication, and cidal will kill. Side means is killing. So when we are talking of virucidal, that means it will kill viruses. Fungicidal, it will kill fungus. Microbicidal, it will kill most of the microorganisms. Sporicidal, it will kill all the spore forms. Tuberculocidal will kill tubercle bacilli. Germicidal, most of the germs will be killed by it. Now, these germicidal and microbicidal are confusing. They can be used, they can replace each other. Microbicidal, microbes, and germs, many of the germs which may be protozoans, and all these will can also be killed by germicidal. So these are certain terms which ends with side, and side is killing. Coming to another term, that is antibiotics. Antibiotics are again chemical or substances produced by one microbe that inhibits or kill another microbe. And these antibiotics can be bacteriostatic as well as bactericidal. And I hope you know the meaning of bacteriostatic and bactericidal by now. Coming to interferons, they are the proteins and they have the property of inhibiting replication of viruses. So they stop multiplication of viruses. Interferons are the ones which stop multiplication of viruses. Antibiotics stop multiplication of bacteria or can kill bacteria too. Coming to a topic that is sterilization. Sterilization, now, since we know what are antiseptics, what are antibiotics, what are interferons, all this. Now coming to sterilize. Sterilization is the term which is used to remove or kill or deactivate all form of viruses, all form of life, not only viruses, all living forms. So, an sterilizing agent will be an agent which will remove all the life forms, total life forms will be removed. It can be done on an article, it can be done on the media. So sterilization, when we are reading in the lab, we are not talking about sterilization in meaning in the other biological forms, or when we are sterilizing human. No, we are not talking of that sterilization. Here we are talking of sterilization which is done in laboratory to sterilize articles or substances that has to be used in the surgery or certain kind of molecular biology laboratory. So that sterilizing agent will be an agent which will remove all living forms. And sterilization is a process of removing all living forms. When we talk about disinfection, it is the process of eliminating all the pathogenic organisms. Now you will say, what is the difference between sterilization and disinfection? In case of disinfection also, we are removing all kind of microorganisms or all kind of living forms. So remember, sterilization is a bigger picture where all kind of living forms will be killed. While when we talk of disinfection, it's usually the removal of pathogenic organisms, that too mostly in their vegetative forms. What is vegetative form? Vegetative form is the growing and multiplying stage of an organism. And when we talk of spore here means the inactive form and in latent state of a microorganism, where whenever it will get a favorable condition, it's going to germinate and then grow and reproduce. So spore form is inactive latent form where the microorganism is waiting for favorable conditions. So when the process of disinfection is done, usually the spore form is not being killed by disinfectants. It in biology exceptions are there, spore forms may be killed, but usually when we are talking of disinfection, then the picture is small. When we are talking of sterilization, it's a larger picture where every life form is removed, while in case of disinfection, usually pathogenic forms which may cause infection are removed, that too mostly in their vegetative forms, rarely in spore form. Decontamination is another word for microbiologists to understand. That is, it's the process of removal of contaminating pathogenic microorganisms. Here we are talking of contaminating because in case of when we are talking of decontamination, it is like there is some organism of interest that may survive. We are decontaminating it. That means we are removing the organisms which we don't need. So decontamination can be achieved by sterilization where we don't need any organism. So we will decontaminate an article or substance from all kind of living forms. We can decontaminate an article or substance by in disinfection also. Disinfection will be removing the pathogenic forms, and decontamination can be done to preserve the organism of interest, and other organisms may not grow in it. So decontamination is a totally different term. It can be achieved by both the process, that is by using disinfectants or by using sterilization. So decontamination is the third kind of process. So there are three, we discussed three processes in this slide. One is sterilization. Sterilization is complete removal of living forms. It can kill, or it can be by killing or deactivating, but it will be complete removal. While disinfection will be removal of pathogenic forms. Most, if we are talking of microorganisms, then mostly vegetative forms will be removed. Spore form may survive in this case. So if we want to perform a surgery, we need a totally sterilized material, and that sterilized material can be obtained by sterilization process, not disinfection. So sterilization can be done by different methods, which I will be talking in the second video. In this video, I just want you to make sure that you are clear with the terms sterilization, disinfection, and decontamination. And I hope you may have understood. And if I'm not clear, I will be very happy to receive comments and improve myself. So please do comment. If you like it, please like it. If you're not disliking it, please let me know what are the points I can improve. Now coming to differentiation between sterilization and disinfection, as I have already talked a lot on it. But if a 3 marks question come in an examination, you may write few things, and these are the points which you can explain and you can elaborate. First, I told you that sterilization is complete removal of living forms, while disinfection is removal of pathogenic forms. They can be in their mostly in their vegetative states if we are talking of microorganisms, rarely in spore form. Please add this line also while you're writing the differentiation between sterilization and disinfection. Then microorganisms are removed in their vegetative as well as spore form, that I have already told you and that I have discussed. So second point is that. Then the various methods that can be applied for sterilization are physical methods, chemical methods, radiation, filtration. While radiation and filtration comes in physical methods itself, but I will be elaborating this radiation and filtration. So I have kept in a different bullet. But while if we talk in a larger term, then physical methods includes radiation and filtration. Then there are two type of disinfectants which can be used, broad spectrum and narrow spectrum. That too I will be discussing in my coming videos. Do stay tuned with me, and if you have any confusions or if I was not able to clear things, please let me know. I will be very happy. Thank you so much.